ಮೇಡಮ್ ಶುರು ಮಾಡೋದ ಮೇಡಮ್?
Yes, sir.
ನಮಸ್ತೆ ಮೇಡಮ್. ಸೊ ಗುಡ್ ಈವ್ನಿಂಗ್ ಎವ್ರಿಬಡಿ. ಸೊ ಇಂದಿನ ಉಪನ್ಯಾಸ ಮಾಲೆಯಲ್ಲಿ ನೇತ್ರಾವತಿ BS ಅವರು ಬಯೋಮಾಲಿಕ್ಯೂಲ್ಸ್ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಅವರ ಬಗ್ಗೆ ಒಂದು ಕಿರು ಪರಿಚಯ ಹಾಗೂ ಅವರನ್ನು ಸ್ವಾಗತಿಸುತ್ತಾ, ಅದೇ ರೀತಿ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಆಯೋಜಿಸಿ ನಮಗೆ ಪ್ರತಿದಿನವೂ ಮಾರ್ಗದರ್ಶನ ನೀಡುತ್ತಾ ಬಂದಿರುವ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ ಅವರಿಗೂ ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಅದೇ ರೀತಿ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರಾದಂತಹ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪ ಸರ್ ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಸೊ ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ನಮ್ಮ ಉಪನ್ಯಾಸ ನೀಡಲಿರುವ ನೇತ್ರಾವತಿ BS ಅವರ ಬಗ್ಗೆ ಒಂದು ಕಿರು ನೋಟ. ನೇತ್ರಾವತಿ BS ಅವರು MSc ಜುವಾಲಜಿ ಸ್ನಾತಕೋತ್ತರ ಪದವಿಯನ್ನು ಬೆಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದಿಂದ ಪಡೆದಿದ್ದು, ಅದೇ ರೀತಿ MPhil ಪದವಿಯನ್ನು ಕೂಡ ಅಣ್ಣಾಮಲೈ ಯೂನಿವರ್ಸಿಟಿಯಿಂದ ಪಡೆದಿದ್ದು, ಕಳೆದ 6 ವರ್ಷಗಳಿಂದ ಆಕ್ಸ್ಫರ್ಡ್ ಪದವಿ ಕಾಲೇಜಿನಲ್ಲಿ JP ನಗರ ಅವರು 2002ರಿಂದ 2008ರವರೆಗೆ ಕರ್ತವ್ಯ ನಿರ್ವಹಿಸಿದ್ದು, ಅದೇ ರೀತಿ ಪದವಿ ಪೂರ್ವ ಶಿಕ್ಷಣ ಇಲಾಖೆ KLE ಕಾಲೇಜ್ ರಾಜಾಜಿನಗರ 2008ರಿಂದ 2013ರವರೆಗೆ ಇಲ್ಲೂ ಕೂಡ ಅವರು 6 ವರ್ಷದ ಸೇವೆ ಸಲ್ಲಿಸಿದ್ದು, ಪ್ರಸ್ತುತವಾಗಿ ಸರ್ಕಾರಿ ಪದವಿ ಪೂರ್ವ ಕಾಲೇಜ್ ತಾಲೂಕ್ ಹಾಸನ್ ಡಿಸ್ಟ್ರಿಕ್ಟ್ ಅಲ್ಲಿ ಕರ್ತವ್ಯ ನಿರ್ವಹಿಸುತ್ತಿದ್ದು, so she is presently working in Government PU College for Girls in Alur, Taluk Hasan district from 2030 till now. Over to you, ma'am.
Thank you, sir. Good evening, one and all. Am I audible?
Audible.
Okay. Good evening, students. Good evening, KSTA. Today, the topic what I'm going to be talking about is biomolecules, which is included in first PUC NCERT syllabus biology under the third unit, cell, the unit of life. Now, when you talk about biomolecules, why are they called as biomolecules? They're called as biomolecules because they are not just molecules, but the molecules which support the living system, which regulate the functions of the living system. So therefore, they are called as the biomolecules. They support the living system. They help the body functioning of the living system. So that is what about biomolecules. Now, the content what we are going to be covering for this 1 hour session is what are biomolecules, the types of biomolecules, the biological significance of biomolecules, what are enzymes and its types, and then we are going to be talking about the cofactors which are related to the biomolecules. Now, let us move on to the first part, that is biomolecules. So as I said, biomolecules are nothing but the chemicals which support the living system. So these are the chemicals which support the living system. They help to protect our body. They are present in our cells. They are present as antibodies. They are present in our digestive tract. They are present in our blood. They are present in as hormones. They regulate the growth of our body. They regulate the reproductive functions of our body. So that is why they are called as biomolecules. Molecules which support the living system. ಯಾವ ಅಣುಗಳು ಒಂದು ಜೀವಿಯನ್ನ ಕಾಪಾಡೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೋ, ಸಪೋರ್ಟ್ ಮಾಡುತ್ತೋ ಅಂತ ಒಂದನ್ನ ಜೈವಿಕ ಅಣುಗಳು ಅಂತ ಕರೀತಾರೆ, ಬಯೋಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ನಾವು ಇದ್ರಲ್ಲಿ ಎಷ್ಟು ವಿಧಗಳಿದೆ ಬಯೋಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಲ್ಲಿ ಮಕ್ಕಳ? 2 ವಿಧಗಳಿದೆ. There are 2 types in the biomolecules, that is the micromolecules and the macromolecules. So ಯಾಕೆ ಈ ತರ ಕರೀತಾರೆ micromolecules? Micromolecules means these have lower molecular weight. They are very small in size. ಅವರ ಗಾತ್ರ ತುಂಬಾ ಚಿಕ್ಕದಾಗಿದ್ದು ಅವರ ಮಾಲಿಕ್ಯುಲರ್ ವೇಟ್ ತುಂಬಾ ಕಡಿಮೆ ಇರೋದ್ರಿಂದ ಅವರನ್ನ ಮೈಕ್ರೋ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಅದರ ತದ್ವಿರುದ್ಧವಾಗಿ ಮ್ಯಾಕ್ರೋ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಂದ್ರೆ ಯಾವ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಗಾತ್ರ ಅತಿ ದೊಡ್ಡದಾಗಿದ್ದು ಅದರ ಮಾಲಿಕ್ಯುಲರ್ ವೇಟ್ ತುಂಬಾ ಜಾಸ್ತಿ ಇರುತ್ತಲ್ಲ ಅಂತ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ನ ಮ್ಯಾಕ್ರೋ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ಮೈಕ್ರೋ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಮೀನ್ಸ್ the molecules which are lower in size with less molecular weight are called as micromolecules. They are water, minerals, amino acids, sugars. These are called as microbiomolecules. Whereas macrobiomolecules means these are the molecules like proteins, carbohydrates, lipids are called as macromolecules or macromolecules. Now, let us move on to the first one, the microbiomolecules. Now, coming on to microbiomolecules, the first is the amino acids. As the name says, amino means these are the molecules which are having nitrogen. They are acidic in nature. These amino acids, there are about 20 naturally occurring amino acids in our body. So amino acids are 20 in number which are present in our body. Coming on to the structure of the amino acids. The amino acids have a central carbon atom, and on the either side they have an amino group and a carboxylic group, and they have a hydrogen group with the down R, which is nothing but the variable group. The variable group can be methane, ethane, any alcohol can be the variable group. So this is the basic structure of an amino acid. So this is very important for the exam, which can be asked for 2 marks, the structure of the amino acid. The structure of the amino acid has a central carbon, on the other side amino group, on one side carboxylic group, on the other side hydrogen and a variable group. The variable group can be ethane, methane, alcohol, or any functional groups can be here in the variable group. Now, when you talk about these amino acids, there are 2 types of amino acids, that is essential amino acids and non-essential amino acids. So what is the meaning of essential? As the name says, essential, very much required for the body. So these essential amino acids are not synthesized by the body. ನಮ್ಮ ದೇಹದಲ್ಲಿ ಅದರ ಉತ್ಪಾದನೆ ಆಗೋದಿಲ್ಲ. ಹಾಗಾಗಿ ನಾವು ಅದನ್ನ ಆಹಾರದ ಮುಖಾಂತರ ತಗೋಬೇಕಾಗುತ್ತೆ. So ಅದನ್ನ essential amino acids ಅಂತ ಕರೀತಾರೆ. The amino acids which have to be taken through the diet are called as essential amino acids. So ಇದಕ್ಕೆ example ಯಾವುದು ಅಂದ್ರೆ leucine, lysine. So these are the examples for essential amino acids. Non-essential amino acids, they are synthesized in the body. ನಮ್ಮ ದೇಹದಲ್ಲೇ ಉತ್ಪಾದನೆ ಆಗುತ್ತೆ. ಹಾಗಾಗಿ ಅವರನ್ನ ಆಹಾರದಿಂದ ತಗೊಳ್ಳೋ ಅವಶ್ಯಕತೆ ಇರೋದಿಲ್ಲ. So they are nothing but alanine, aspartic acid. So these are the 2 types of amino acids, essential amino acids and non-essential amino acids. Essential amino acids are leucine, example. Non-essential amino acids are alanine or aspartic acid. Now, let us move on to the next concept. How are these amino acids linked? Now, these amino acids, they form a macromolecule which is called as the proteins. So proteins are the macromolecules. These macromolecules are made up of basic elements carbon, hydrogen, oxygen, and nitrogen. So these are the basic components or the elements present in the protein. In turn, these molecules are going to bond with each other and form the subunits. The subunits of the proteins are amino acids. So proteins are bound to each other by amino acids. So there are many amino acids which are linked together to form one chain of protein. Now, let us understand how these amino acids are linked together to form a protein. How is it? So, the bond which links the amino acid is called as the peptide bond. How is the bond linked? Let us see that. Now you have the carbon, you have the carboxylic group on one side, the amino group on other side, the hydrogen and the variable group. At the same time, you have another amino acid where you have an amino group, a carbon, and you have a carboxylic group and hydrogen and a variable group. Now, how does the bonding occur? The bonding is going to be between the hydroxylic group and the hydrogen group of the carboxylic group and the amino group. Once again, I'm going to repeat. The peptide bond is a bond between the carboxylic group of one amino acid and the amino group of another amino acid with the elimination of a water molecule. So one water molecule is going to be released to link two amino acids. So this bond is called as the peptide bond, which will be asked for 1 mark. What is peptide bond? It is a bond between two amino acids where the carboxylic group of one amino acid is linked with the amino group of another amino acid with the release of a water molecule, and that forms a peptide bond. Let us move on to the next concept, the structure of proteins. How is the structure of proteins? So when you talk about the structure of proteins, there are four different structures of proteins. One is primary structure, secondary structure, tertiary structure, and quaternary structure. Now, coming on to primary structure, as the name says, the structure is very simple. That means it is linear with one side amino group and the other side carboxylic group. It is a linear chain. There are no folds. The amino acids are arranged in a linear manner. The example for that is insulin. Now, coming on to the secondary structure. The secondary structure is little bit complicated with the helical structure like this. One side amino group, on the other side carboxylic group. So it is little complicated when compared to the primary structure. So for that, the example is the myosin. So I hope you know what is myosin. Myosin is the contractile protein present in the muscle fibers. So you know what is insulin? Insulin is the hormone which controls the blood sugar. Next, we have the tertiary structure. The tertiary structure is more complicated than the secondary structure, which is folded in the form of a sphere, like a woolen ball. So that is the tertiary structure. So the tertiary structure is more complicated when compared to the secondary structure. So for that, the example is the myoglobin. So myoglobin is very much similar to hemoglobin, but it is present in the muscle fibers. In the muscle cells, we find the myoglobin, which helps to absorb the oxygen and give out the carbon dioxide. The last one is the quaternary structure. The quaternary structure, I'll write on the top, the quaternary structure is very complicated, which has many loops like this. There are a lot of loops in the quaternary structure, and all of them are linked to a non-protein part. So this is how the quaternary structure looks. The quaternary structure example is hemoglobin, which is present in RBC cells. So that is the structure of hemoglobin, which is a quaternary structure. Now, let me repeat once. The structure of the protein is primary, which is linear, secondary, which is spring-shaped, tertiary, which is complicated and looped. The quaternary is highly complicated with many folds, and the example for that is hemoglobin of RBC. Let us move on to the next part, the biological significance of the proteins. What is the biological significance of proteins? So when you talk about the biological significance of proteins, the significance of proteins include they form the structural component of the cell. They form the structural component. As you have heard in your first PUC, you know about areolar connective tissue, adipose connective tissue, in which there are white and yellow fibers. So the white fibers have collagen protein, which is a protein, and the yellow fibers have elastin protein. So proteins form the structural component of the cell. Next, the proteins form the components of blood. That is, you have the immunoglobulins. So proteins form the component of the blood as immunoglobulins, which protect our body from antigens. Then we have the blood clotting proteins. Clotting factors are there, which are also the proteins, which are present in the blood to help in clotting mechanism. Protein also act as enzymes, which I'm going to discuss in the later part of the class. They act as enzymes to catalyze the reaction. Proteins also occur as hormones. There are a lot of hormones in our body, like growth hormone, like thyroxine hormone. So these hormones are all nothing but proteins. So all proteins are hormones. Okay, almost all the hormones are proteins. So proteins act as structural component. Proteins are present in blood as immunoglobulins and clotting factors. Proteins act as enzymes, which are helping in digestion and other activities. Proteins are as hormones. So there are a lot of activities performed by the proteins. Therefore, the proteins are called as body builders. They build our body. The body or the entire body is made up of proteins. Proteins are also present on our skin as melanin. Proteins are also present in our hair as keratin. So all these are nothing but proteins. So you have lot of proteins in our body. So therefore, proteins are called as body builders. Let us move on to the next part of the biomolecule. We are finished with the proteins. Let us move on to the next biomolecule, that is the sugars. Sugars are the micromolecules which are lower sized and with lesser molecular weight. Now, when you talk about sugars, sugars are common, these are scientifically called as saccharides. Chemically called as saccharides, and commonly we call them as sugars. Now, these sugars, they are classified based on the carbon atom. How many carbon atoms they have? So based on that, you have different types of sugars, that is hexose, pentose, there are not triose, tetrose and all that. But the most significant ones are hexose and pentose. The hexose are the ones which we have when we fall down, when we are tired, we have that. That is nothing but the glucose. So glucose is a sugar which is having 6 carbon atoms. Then you have the sugar in fruits, that is fructose. The sugar in milk, that is called as galactose. So these are the sugars which are hexose. Then we have pentose. The pentose sugars have 5 carbon atoms. They are present in DNA and RNA as deoxyribose and ribose. So sugars are also called as saccharides, and they are classified into many types, among which 2 are very important, that is hexose and pentose. Hexose has 6 carbon atoms, pentose has 5 carbon atoms. The hexose are glucose, fructose and galactose. The pentose are deoxyribose and ribose, present in DNA and RNA respectively. Now let us move on to the macromolecule, that is carbohydrates. Now, when you talk about carbohydrates, so carbohydrates, as the name says, carbohydrates, they are made up of carbon, hydrogen and oxygen. The carbohydrates are also called as polysaccharides. The carbohydrates are also called as polysaccharides, and they are made up of the sugars. The carbohydrates are the macromolecules which are linked by the sugar molecules. Now, when you talk about these carbohydrates, they are linked by a bond called as glycoside bond. That is, an oxygen molecule links the 2 sugar molecules. Now, coming on to the types of carbohydrates. There are 2 types of carbohydrates, that is homopolysaccharides and heteropolysaccharides. Homopolysaccharides, as the name says, the polysaccharides which are having same type of sugar molecules are called as homopolysaccharides. Example for homopolysaccharide is starch and cellulose and glycogen. So these are the examples of homopolysaccharides. Homopolysaccharides means the polysaccharides which have same type of monomers, whereas heteropolysaccharides means these have more than 2 types of monomers to form a heteropolysaccharide. Example for that is chitin, inulin. So chitin is made up of glucose and galactose. Inulin is made up of glucose and fructose, whereas starch is made up of only glucose. So homopolysaccharides means the saccharides which are made up of only 1 monomer. Heteropolysaccharides means the saccharides which are made up of more than 2 types of monomers. This will be asked for the exam for 2 marks as a difference. Differentiate between homopolysaccharides and heteropolysaccharides. So homopolysaccharides means the saccharides, the carbohydrates which have only 1 type of monomer are called as homopolysaccharides. The heteropolysaccharides means the saccharides which are having more than 2 types of monomers. The example for homopolysaccharide is starch, cellulose and glycogen. The example for heteropolysaccharide is chitin and inulin. Let us move on to the biological significance of the carbohydrates. Now, coming on to the biological significance of carbohydrates. Now, when you talk about the biological significance of carbohydrates, carbohydrates are very famously called as metabolic fuels. Why are they called as metabolic fuels? Because they give instant energy to the cells. They provide energy to the cells to perform their activities. So that is why they are called as metabolic fuels. Carbohydrates na metabolic fuels anta yaake karitare andre because ಅವರು ಇನ್ಸ್ಟೆಂಟ್ ಆಗಿ ಎನರ್ಜಿ ಕೊಡ್ತಾರೆ. ಯಾರ ನಾವು ಬಿದ್ದಾಗ ನಾವು ಫಸ್ಟ್ ಏನ್ ತಗೊಂತೀವಿ? ಗ್ಲೂಕೋಸ್ ತಗೊಂತೀವಿ. ಯಾಕೆ ಅಂದ್ರೆ ಅದರಿಂದ ಇನ್ಸ್ಟೆಂಟ್ ಎನರ್ಜಿ ಬರುತ್ತೆ. ಬೇಗ ಸೆಲ್ಸ್ ಅದನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡ್ಕೊಂಡು ಬೇಗ ಎನರ್ಜಿ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. ಹಾಗಾಗಿ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್ ನ ಕಾಮನ್ ಆಗಿ ಏನಂತ ಕರಿತೀವಿ? ಮೆಟಬಾಲಿಕ್ ಫ್ಯೂಯಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ದೇ ಗಿವ್ ಇನ್ಸ್ಟೆಂಟ್ ಎನರ್ಜಿ ಟು ದ ಸೆಲ್ಸ್. Now coming on to carbohydrates, they form structural component of the cell. How? Now, when you talk about the cellulose, so we all know that the cell wall of the plant cell wall is made up of cellulose. At the same time, they also form, you know about chitin. So arthropods have the outer exoskeleton, which is made up of chitin. So they form the structural component of the cell or organisms, where the cellulose is seen in plant cell wall and chitin is seen in the exoskeleton of arthropods. Next, the carbohydrates also are present as storage component. They are used as storage component. That means the starch is present as reserve food material in plants and the glycogen is present as reserved food material in animals. So starch is the reserve food material of plants and glycogen is the reserve food material of animals. The starch is stored either in the root or in the stem or in the leaf of the plant. The glycogen is stored in the muscle and under the skin of the animals. Next, the carbohydrates are also of economical importance. How are they economically important? Because the carbohydrates are being used extensively to prepare ropes, to prepare papers. So for that, we are cutting the plants and the plant is nothing but made up of cellulose. So they are very economically important to humans also. So carbohydrates are called as metabolic fuels. They are the structural component of cellulose, that is, and chitin. They also act as storage components like starch and glycogen, and they are also economically important to humans as we are using it to prepare paper and other products. Let us move on to the next macromolecule. The next macromolecule is the lipids. Coming on to lipids. So lipids are also commonly called as fats. So we call them as fats. Now these fats or lipids are made up of what is called as fatty acids and glycerol. So the subunits of lipids are fatty acids and glycerol. Now, these lipids, we all know that it is insoluble in water, but soluble in alcohol. Okay, so lipids are otherwise called as fats. The subunits of lipids are fatty acids and glycerol. They are insoluble in water, but soluble in alcohol. Now, very important in lipids is the significance of lipids, which will be asked for 3 marks in the exam. That is the biological significance of lipids. Now, when you talk about the biological significance of lipids, so lipids play a very important role as they are found around the organs as protective layer. So they act as protective layer around the organs. They also act as shock absorbers. They absorb the shock which is experienced by our organs. So whatever the organs absorb, the jerks, what the organs take up is all going to be absorbed by the lipids. And then they are the food provider. So when we undergo starvation, when the organism undergoes starvation or fasting, at that time, the lipids are going to be utilized by the cells to provide energy. So they act as food providers or energy providers. Then the lipids are found around. They are also found around the fur, the feather, and leaves of organisms. They are found around the fur. The polar bear's fur is lined by the lipid layer. The feathers of the birds are lined by the lipid layer. The leaves of the plants are lined by lipid layer to protect them from the harsh conditions. Then the lipids are also found around the neurons as myelin sheath. So neurons are covered by myelin sheath, which helps in faster conduction of the impulses. So what is the meaning of impulse? Message. So messages are going to be passed on by the neurons if they have the myelin sheath. And we all know that the neurons are having myelin sheath. They are called as myelinated neurons. So these myelinated neurons will conduct the impulse faster when compared to the neurons without the myelin sheath. So these are the important significance of the lipids. Once again, the lipids form the protective layer around the organs. It is also found beneath your skin to protect our body, and a very important thing, it acts as insulators. It protects the body from the cold conditions. It protects our body from cold conditions. It acts as shock absorber. Lipids are present covering the heart as pericardium, discovering the lungs as pleura, and it is also covering the kidney as the renal capsule. So all these are protecting the important organs of our body as shock absorber. The lipids are stored in the adipose tissue, which provide food during starvation. And the lipids are present around the neurons as myelin sheath, which will help in faster conduction of the nerve impulse. And the lipids protect the human body from the cold and they act as insulators. So these are the important functions of the lipids which are present in our body. Now, let us go on to the other important macromolecule, that is nothing but nucleic acids. Now, when you talk about nucleic acids, so as the name says, nucleic acids. So as the name says, we get to know that it is present inside the nucleus of a cell. It is present inside the nucleus of a cell and it is acidic in nature. The nucleic acids were first discovered or first noticed by Michel. So Friedrich Michel was the scientist who was the first to see these nucleic acids inside the cell and named it as nuclein. But later on, another scientist by name Altman, he renamed the nucleins as nucleic acid. So the nucleic acids were first discovered by Friedrich Michel, and he called it as nuclein. But another scientist by name Altman, he got to know that these nucleins are acidic in nature because they have a phosphate group in them. So therefore, he renamed it as nucleic acids. Now, let us move on to the chemical composition of the nucleic acid. What is there in the nucleic acid? Now, when you talk about the nucleic acid is made up of what is called as, it is made up of many nucleotides. So there are polynucleotides which form one nucleic acid. Now these nucleotides, each nucleotide is made up of two components. That is the nucleoside and the phosphate group. The nucleoside is further made up of two components, that is the nitrogenous base and the pentose sugar, what we discussed under carbohydrates. The phosphate is nothing but orthophosphoric acid. Which is nothing but H3PO4. So this is the group which is giving acidic nature to the nucleic acid. Now when you talk about the nucleotides, so this is very important for the exam. The nucleotides are nothing but, they are made up of two components, nucleosides and phosphate. The nucleoside is further made up of nitrogenous base and pentose sugar. The nitrogenous base are of two types, purines and pyrimidines. Whereas the pentose sugar are of two types, that is, there are two different types of pentose sugar, that is deoxyribose and ribose. So this is the overall chemical composition of the nucleic acid. Now when you talk about the purines and pyrimidines, the purines are nothing but adenine and guanine. The pyrimidines are cytosine, uracil and thymine. So these are the nitrogenous bases present in each nucleotide. Once again, I'm going to repeat. The nucleic acids are made up of many nucleotides or polynucleotides. Each nucleotide composes of nucleoside and a phosphate. The nucleoside comprises of nitrogenous base and pentose sugar. The nitrogenous bases are of two types, purines and pyrimidines. There is structural difference between the purines and the pyrimidines. The purines have double benzene ring, whereas the pyrimidines have single benzene ring. Not much detail required in biology. Now, when you talk about purines, there are two types of purines, adenine, which is represented as A, and guanine, which is represented as G. And pyrimidines are of three types, cytosine, represented as C, uracil, represented as U, and thymine, represented as T. Now, when you talk about the pentose sugar, you have the deoxyribose in DNA and the ribose in RNA. The phosphate group remains the same, orthophosphoric acid, which gives acidic nature to the nucleic acids. Let us go on to the next part, that is the types of nucleic acids. Now, coming on to the types of nucleic acids, there are two types of nucleic acids, that is DNA and RNA. So let us understand what are the differences between the DNA and the RNA. Now, when you talk about the DNA, so DNA means deoxyribose nucleic acid and RNA means ribonucleic acid. Now, when you see the chemical content in the DNA is made up of deoxyribose sugar and the RNA is made up of ribose sugar. The DNA is the DNA is having adenine, guanine, cytosine, and thymine. Whereas the RNA is having adenine, guanine, cytosine, and uracil. So instead of thymine, the RNA is having uracil. So these two are the chemical difference between DNA and RNA. Let us understand the structural difference between DNA and RNA. The DNA is helical in structure. So the DNA is helical in structure, whereas the RNA is single. It is single-stranded. It is made up of only one strand with some loops. So the DNA is helical in nature, and whereas the RNA is single. Now, when you talk about the DNA forms the genetic material in majority of cells. So, in majority of cells, the DNA is the genetic material, whereas RNA is the genetic material only in viruses. So, only in viruses we can see RNA as the genetic material, whereas it undergoes what is the process called as protein synthesis. Now, coming on to DNA. DNA plays a very important role in evolution. It helps in passing the characters from one generation to another generation, and it helps in leading for variations. So whatever the variations which we see in offsprings or from generation to generation is because of this DNA, whereas RNA is genetic material only in very few organisms, like the coronavirus has RNA as a genetic material, the HIV virus has RNA as a genetic material. So it is genetic material only in few organisms, whereas it helps in protein synthesis. So the proteins which are produced in our body is all synthesized inside this RNA or by this RNA. So this is the major difference between DNA and RNA. There are the more differences, but in order to understand it briefly, so DNA is made up, DNA is called as deoxyribonucleic acid, RNA is called as ribonucleic acid. DNA is made up of a pentose sugar, deoxyribose. RNA is made up of a pentose sugar, ribose. The DNA has purines, adenine and guanine, pyrimidines, cytosine and thymine, whereas RNA has adenine and guanine as purines, pyrimidines are cytosine and uracil. So in RNA, instead of thymine, we have uracil. Now, the structure of DNA is helical. As we know, Watson and Crick proposed the double helix model. So according to them, the DNA is helical in shape, whereas RNA is made up of only one single strand. The DNA is the genetic material in majority of organisms. It helps in variations. It also leads to evolution. Whereas the RNA is the genetic material in very few organisms, that is nothing but the viruses. And remember, the viruses which have RNA as a genetic material are called as retroviruses. So corona comes under retroviruses. What is the function of RNA? It helps in protein synthesis. Now, I hope you've understood about the difference between the DNA and the RNA. Now, DNA is of only one type. There are no types of DNA, but the RNA is of three types on a functional basis. So, RNA is of three types, that is tRNA, rRNA, and mRNA. Okay, so tRNA, transfer RNA. So it transfers the amino acid towards the site of the protein synthesis. Whereas rRNA means ribosomal RNA. This will help in protein synthesis. This is the factory where actually the protein is going to be synthesized in the cell. The last one is mRNA, messenger RNA. This is the one which transfers the message from DNA into a protein. So this concepts you're going to be understanding in the higher classes, but just know that the RNA is of three types: tRNA, which is called as transfer RNA, rRNA, which is called as the ribosomal RNA, and mRNA, which is called as a messenger RNA. So that's about the nucleic acids. So let us move on to the next content of this topic, that is enzymes. Very important. Okay, now coming on to enzymes, what are these enzymes? For example, if you want to understand the function of enzymes, let me give you an example. For example, if you have to look for a rented house, what do? We don't go directly. We look for a mediator. We look for a broker to look for a house for rent. In the same way, in our body, for all the chemical reactions to take place, there is an enzyme which is going to mediate the reaction and minimize the energy loss. Okay, so enzymes are called as biocatalyst. So biocatalyst means these are the brokers or the catalyst who is going to take part in our reactions. So all the metabolic reactions which takes place in our body is mediated by the enzymes. So enzymes play a very important role in all the chemical reactions. So what do? They increase the rate of reaction. So they make the reaction to happen faster. They accelerate the rate of reaction with minimum energy utilization. So kadme energy use ಮಾಡ್ಕೊಂಡು ಬೇಗ ಪ್ರಾಡಕ್ಟ್ಸ್ ಆಗೋದಕ್ಕೆ ಸಹಾಯ ಮಾಡೋದು ಯಾವುದು ಅಂದ್ರೆ enzymes. ಅದಕ್ಕೆ enzymes ನ biocatalyst ಅಂತ ಕರೀತಾರೆ. Chemically ಏನಿದು enzymes? Enzymes are proteins. So there is a saying that all enzymes are proteins, but all proteins are not enzymes. ಎಲ್ಲ enzymes protein, ಆದರೆ ಎಲ್ಲ protein enzyme ಅಲ್ಲ ಅನ್ನೋದು ಒಂದು word ಇದೆ. Now when you talk about these enzymes, enzymes are very specific about their pH in the body. The body pH variation ಆದರೆ enzyme ಕೆಲಸ ಮಾಡೋದಿಲ್ಲ. They are very specific about temperature. So that is why when we get fever or when our body is tired, we don't feel the taste of the food because our enzymes are going to be inactive. Now, enzymes are very specific about the substrate. Now, what is the meaning of substrate? ಯಾವದ್ರ ಮೇಲೆ enzyme ಕೆಲಸ ಮಾಡುತ್ತೆ, on which part or on which object the enzymes are going to act. So that is called as a substrate. Protein ಮೇಲೆ act ಮಾಡುತ್ತಾ, carbohydrates ಮೇಲೆ act ಮಾಡುತ್ತಾ, lipids ಮೇಲೆ act ಮಾಡುತ್ತೋ. So these are called as the substrate on which the enzymes are going to act upon, called as substrate. So enzymes which act upon protein will not be acting on carbohydrates. So they are very specific about the pH, they are very specific about the temperature and about the substrate. Now where actually the enzymatic reactions takes place? The enzymatic reactions takes place in regions called as active sites. There are active sites where the enzymatic reactions are going to take place. So these are the regions where the enzyme is going to convert the substrate plus enzyme will give rise to enzyme substrate complex, and later this will again form into product and you'll get back your enzyme. Okay, so this is how the enzymes act. The enzymes are going to combine with the substrate. They form a complex called as enzyme substrate complex. And again, these enzymes are going to complete the reaction where you get back your enzyme with the product. So that is how the enzymes are going to react in our body. That is the mechanism of enzyme action. Next, moving on to Srinivas sir, continue ಮಾಡ್ಬೋದಾ?
Madam, continue, please continue.
Okay. Now let us move on to the next part, that is types of enzymes. How many types of enzymes are there? There are different types of enzymes. So I hope you all know about IUPAC. So IUPAC is It is an organization which takes care about the chemical coding and the naming of the chemicals. Now, this IUPAC has given the different types of the enzymes based on a criteria, that is based on the reaction they catalyze. So based on the reaction they catalyze. So what is a reaction they catalyze? So based on that, you have different types of enzymes. The first enzyme is Oxidor Reductase. So what is Oxidor Reductase? Oxidor Reductase, as the name says, these are the enzymes which help in oxidation and reduction. So oxidation and reduction reactions are catalyzed in the body by Oxidor Reductase enzymes. Next we have hydrolase. So these are the enzymes which help in hydrolysis. They help in hydrolysis. So all the digestive enzymes are called as hydrolase because for the hydrolysis to take place, water is a very important component. So all the reactions which takes place in the presence of water are called as hydrolysis. So hydrolase enzymes help in hydrolysis. So all the digestive reactions which takes place in the body are all done by hydrolase enzymes. The next one is lyase. So as the name says, lyase, to break down. So these are the enzymes which break the compound. So it breaks a complex compound into a simpler molecule. So that is the main function of lyase. So it breaks a big compound to form the smaller products. So that is the main function of lyase. So they help in what is called as lysis. The reaction is called as lysis. The next one is ligase. So this is opposite to lyase. The ligase enzyme helps in joining of components. That is, it helps in ligation. Ligation means it joins two components to form one big compound. That is the main function of ligase. And we have the last one, that is isomerase. So what is isomerase? As the name says, isomerase. So this helps in isomerization. What is isomerization? That means one form of compound will be converted into another form of compound. So there are different forms of compound. So the isomerase converts one form of compound into another form. For example, glucose 6-phosphate will be converted into fructose 6-phosphate. So that is what is called as isomerization. And the enzymes which take part in isomerization are called as isomerase. So this is a very important question which is going to be asked in the exam for 5 marks. That is, they're going to ask, classify the enzymes based on the type of reaction they catalyze. That is, Oxidor Reductase, which takes part in oxidation and reduction, hydrolase, which takes part in hydrolysis, lyase, which takes part in lysis, ligase, which takes part in ligation, and isomerase, which takes part in isomerization. So this is a very important question which will be always asked for first PU students in their final question paper. So I hope you've understood about the types of enzymes. Right. So let us go on to the last part of the session, that is cofactors. So what are these cofactors? Cofactors means these are nothing but the factors which are present attached with the proteins. So these are called as apofactor or apoenzyme. They're called as apofactor or apoenzyme. So the protein part will not be always only protein. The protein will be associated with either a pigment or a metal ion. So these are called as the cofactors. The non-protein part of the component is called as the cofactors or apoenzyme. Now what is the protein part called as? The protein part is called as coenzyme. So for example, if you have heard of hemoglobin, or in hemoglobin, you have protein part and you have the non-protein part. For example, the non-protein part is the pigment, heme part, that is iron part, and that is associated with the globin. So like that, what are cofactors means? These are the factors which are associated with protein for normal functioning of the molecules. So one is that heme part, and the other one is the carbonic anhydrase. So what is the carbonic anhydrase? So this is an enzyme which is present in the blood, in the lungs. So these enzymes, what they do is they help in conversion of carbon dioxide, transport of carbon dioxide. So these enzymes are associated with zinc. So it is a protein, but this protein part is associated with a metal ion, that is the zinc. So the zinc forms the upper part or the cofactor, and the carbonic anhydrase forms the protein part. So like that, the proteins are associated with non-protein part like pigment or the metal ions, which act as cofactors and help in the functioning of the proteins. So this is about the biomolecules chapter. I have concised the entire content to 1 hour. So we all know this is a very brief part of biomolecules chapter. I hope I have explained the important points of biomolecules. Let me just recap what we have done in this one session. So we discussed what are biomolecules, what are the types of biomolecules, that is micromolecules and macromolecules. We discussed what are amino acids, the structure of amino acid, the types of amino acids, then proteins. What are proteins? The structure of proteins, that is primary, secondary, tertiary, and quaternary. Then the biological significance of proteins. Then we moved on to carbohydrates, that is sugars. So what are sugars? What are the types of sugars? What are carbohydrates? How many types? Homopolysaccharides and heteropolysaccharides. Then the biological significance of the carbohydrates. Then we moved on to lipids, its biological significance. Then nucleic acids. So what are nucleic acids? Chemical composition of nucleic acids, types of nucleic acids, that is DNA and RNA. Then we also discussed the important difference between DNA and RNA. Then we talked about enzymes, the properties of enzymes, and the classification of enzymes according to the reaction what they catalyze. And last, we discussed about cofactors. I hope I have made you understand about biomolecules in a very brief manner or in a shared manner. Thank you, sir, for giving an opportunity to talk about biomolecules. I hope I have done it perfectly.
It was excellent. You covered everything about biochemistry in a very nutshell. Your students would got a lot of take-home message from your talk. You biochemistry ಇದೆಲ್ಲ ಕವರ್ ಮಾಡ್ಬಿಟ್ರಿ. Excellent. ಇನ್ನು ಏನಾದರೂ ಡೀಟೇಲ್ ಆಗಿ ಹೇಳೋಂಗಿದ್ರೆ we can arrange one more talk. You can take a topic and suggest us.
Yes, sir. Yes, sir.
I studied biochemistry very long ago.
Yes, sir.
Bachelor's and masters. There recap.
Yes, sir.
Keep it up.
Thank you, sir. Thank you very much, sir. Thank you.
Other talk, please suggest. That should be of use for second year PUs.
Yes, sir. Thank you, sir. Thank you. Thank you, sir. Thank you.
Students,
Any
yes,
doubts?
really very good. Madam Netrvathi, madam. Sure, most of you have understood what are biomolecules. Madam will be giving one more talk on one topic of interest for you in the coming days. For now, thank you very much. Over to Siddhagowda for tomorrow's talk. You can the title of the talk. Thank you, sir. Thank you, madam.
So thank you, Ramesh sir. Thank you, KSTA. Thank you, one and all, to give me an opportunity to talk about biomolecules. Thank you, sir. Thank you.
Thank you, madam.
Thank you, sir. Thank you.